Okay, hello everyone. Thank you so much for joining our webinar today, Whole Genome Sequencing for Cronobacter and Other Pathogens in Infant Formula. I'm very excited to have Keren Breiterman and Andrzej Benkowski here with me today to host the webinar. I am Ellie Abraham and I will be moderating today's session. Today's presentation will be about 30 minutes. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer viewers' submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit Enter on your keyboard. Remember, you can submit questions throughout the webinar. That concludes my brief introduction. Keren, I will now pass off the webinar to you for your introduction. You may begin your presentation.
Thanks, Ellie, for that introduction, and thank you everyone for joining this webinar today. My name is Keren Breiterman, and I'm one of the Senior Business Development Directors here at Eurofins North America, and my subject matter expertise is infant formula. So I hope that I'm a familiar name for most of you already. But for those who don't know me, I've been in the third-party testing space for about 16 years now, and I started originally in New Zealand and Singapore, serving the Southeast Asia market, and I've been with Eurofins in the US for the last 8 years. I have a deep passion for the infant formula industry, as I think what you're doing is so important and so critical. And I love that you've chosen Eurofins to partner with you for your food safety needs, whether it be analytical, microbiological, or in any other capacity. It's really an honor to be working with you all. So thank you very much. I also became a mother for the first time last year, as my daughter was born right into the shortage that happened last year. So this is all very personal now as a consumer or as a buyer of infant formula. So with that, I will turn off my camera just to make sure there's no connectivity issues. But there we go. Perfect. So why are we here today? Here we go. Perfect. Thanks, Andrzej. So the infant formula shortage of 2022, as I'm sure you're all aware, started off by supply chain issues carrying over from the COVID-19 pandemic, but then really exacerbated and was thrown into the national headlines by a plant shutdown in early 2022 due to safety concerns over microbiological contamination, specifically Cronobacter. Since then, we've seen actually multiple manufacturers issue voluntary recalls because of Cronobacter contamination. So, you know, it's a widespread issue, or it's becoming a very widespread issue. Cronobacter is such a ubiquitous organism in the infant formula manufacturing environment, and we know how important it is for you to control and mitigate its presence so it doesn't end up in final product where it can have detrimental effects on the consumer. In this case, infants, yeah, it's not a good thing when an infant consumes Cronobacter as opposed to an adult, right? So we understand its importance. So the FDA, in response to the shutdown and the recalls last year, issued guidance in March 2023 regarding the mitigation of Cronobacter and Salmonella in infant formula production environments. And part of the guidance includes a strong recommendation to implement whole genome sequencing techniques as part of root cause investigation. So that is when Cronobacter or Salmonella is confirmed as detected in either an environmental sample or in a product sample, that the manufacturer take the isolate through to whole genome sequencing in order to identify the strain of Cronobacter or Salmonella in order to provide a more complete picture for closing out the investigation and getting to a root cause in order to prevent the pathogen from re-entering the environment. So this is just some of the wordings from that guidance that the FDA issued. So Eurofins, in our efforts to support this vital industry and these recommendations from the agency, has an offering for whole genome sequencing through our microbiology lab in Madison, Wisconsin. My colleague Andrzej Benkowski is here to talk in more detail about whole genome sequencing in general, how powerful a tool it can be for you as manufacturers, and our offering specifically here at Eurofins. So with that, I'll hand the floor over to Andrzej, and I'll be here to help with any Q&A at the end. Andrzej, over to you.
Thanks, Keren. Hi, everyone. Thanks for tuning in today. As Keren said, my name is Andrzej Benkowski. I'm the technical manager for probiotics dietary supplements based out of the Eurofins microbiology laboratory in Madison, Wisconsin. I've been working in the industry for about 15 years, and I also manage our genomics service offering also here in Madison. So today, I'm going to talk about a number of things, starting with a brief overview of the concept and technology of whole genome sequencing. Next, I'm going to discuss the benefits of whole genome sequencing for the identification of pathogens such as Cronobacter. And then finally, I'm going to describe a little bit about our whole genome sequencing or next generation sequencing service offering and the different ways that we utilize the technology for the identification of bacteria here in Madison. All right, so first, let's dive into what whole genome sequencing is as a technology. So let's start off with some terms and definitions. Whole genome sequencing, or WGS, is a comprehensive method for determining the number and order of nucleotides that make up an organism's genome. A genome is a complete set of genes or genetic material present in a cell or organism and defines a living thing by the structure of and order of its nucleotides. The advent of next generation sequencing, or NGS, has allowed the use of whole genome sequencing to become much faster and much cheaper, and has the ability to sequence large amounts of DNA very quickly. It can be used to sequence entire genomes, exomes, or targeted regions of DNA. Has a wide range of applications, such as medicine, agricultural, things like that. And then we have something called long read sequencing. And this is sequencing that can produce reads that are up to 100,000 base pairs long. Alternatively, there is a technology also called short read sequencing. And the main difference between the two, obviously, is the length of reads that they produce. Short read sequencing can only produce reads that are a few hundred bases long. And this difference in read length does have a number of implications for the applications of each technology. So long read sequencing is better for assembling genomes, identifying structural variants such as deletions and insertions. And this is because long reads can span across these variants, while short reads can only see the individual pieces of a variant. And long read sequencing is also better for studying repetitive regions of the genome, such as telomeres or centromeres. Short read sequencing, on the other hand, is better for identifying point mutations, such as single nucleotide polymorphisms, or SNPs. And this is because short reads have higher accuracy than long reads. It's also better for studying gene expression and can be used to measure the levels of different genes in a sample. In general, long read sequencing is better for understanding the structure of the genome, while short read sequencing is better for understanding the function of the genome. So getting into a little bit of the history of whole genome sequencing, it can be traced back to the early 1970s when a scientist named Frederick Sanger developed a method for sequencing DNA. This method, known as Sanger sequencing, is still used today, but it is much slower and much more expensive than the newer sequencing technologies. In the early 1990s, the Human Genome Project, or HGP, was launched. The goal of the HGP was to sequence the entire human genome. This was a massive undertaking and was completed in 2003, sequenced with the first draft being published in the year 2000. This was a huge breakthrough in the field of genetics. Shortly after the human genome was sequenced, the first next generation sequencing technology was developed, and these technologies were at the time and still are much faster and cheaper than Sanger sequencing, and therefore they have made whole genome sequencing much more accessible to researchers and clinicians. So along with the development of these next generation technologies, we saw the cost of sequencing drop dramatically. So here we have a chart of the cost per human genome, and as you can see, around the turn of the 21st century, the cost to sequence a human genome was $100 million, tens of millions of dollars. And with the advent of next generation sequencing, that price has reduced significantly to only around $1,000 today. Because the human genome was sequenced by Sanger sequencing was one of the main reasons why it was so expensive. Some other reasons that we saw a great decrease in the cost of sequencing is an increased competition in the sequencing market. So there are now a number of different companies that offer WGS services, and that's led to increased competition and lower prices, as well as government funding. So governments have invested heavily in WGS research, which has also helped to drive down the costs. This has led to a number of new applications. This reduced costs, such as personalized medicine, disease diagnosis, and things like ancestry tracing. As the cost of WGS continues to decrease, it's likely that this technology will become even more widely used in the future, and it does have the potential to revolutionize the way we diagnose and treat diseases. It can also help us better understand the human genome and its role in human evolution. So as you can see on this slide, there are many different next generation sequencing brands available on the market. Some of those popular include Illumina, Thermo Fisher, Oxford Nanopore Technologies, Pacific Biosciences, 10x Genomics, BGI Group, QIAGEN, Roche, Abbott, Life Technologies. There are many options available, and each have their advantages and disadvantages. So some of the most popular brands include Illumina. This is one of the leading providers of NGS platforms. They're known for their high throughput and accuracy with their short length technology. However, they can be expensive. Thermo Fisher Scientific, another leading provider of NGS platforms. They offer a good balance of price and performance. Oxford Nanopore Technologies, this is a relatively new entrant into the NGS market. ONT platforms offer long reads, which are useful for applications such as de novo genome assembly. However, they can be less accurate than other platforms. Pacific Biosciences is another provider of long-read NGS platforms. They're known for their accuracy. However, they can be expensive and difficult to use. 10x Genomics is a provider of single-cell NGS platforms. They are known for their ability to sequence single cells, which is useful for applications such as cancer research. However, they can be expensive as well. These are just a few of the many different brands on the market. So it's important to know if you're going into this field, you need to know what your specific needs and budget are and base that on the type of instrument that you want to use for sequencing. So here at the Madison Laboratory, we have an Oxford Nanopore GridION, and we use Oxford Nanopore technology long-read sequencing. So I wanted to discuss a little bit more about this technology and how it works. This is a third-generation sequencing technology. It uses a protein nanopore to sequence the DNA. So the nanopore is this tiny hole in a membrane that allows single-stranded DNA to pass through it. As the DNA passes through the nanopore, it's affected by an electric field, which causes it to change the current that flows through the pore. The changes in this current then are detected by a sensor, and the data is used to determine the sequence of DNA. As I mentioned earlier, this sequencing technology can produce reads that are up to 100,000 base pairs long, which is much longer than the reads that can be produced by other sequencing technologies. And this makes it a valuable tool for studying the structure of genomes, including repetitive regions and structural variants. It's still a relatively new technology, but it does have the potential to revolutionize the way we study the genome. It's already being used for a variety of applications. Here in our lab, we have a lot of different practical uses for it, but also in the research and academic space, it's being used for genome assembly, structural variant detection, epigenomics, metagenomics, single-cell sequencing. Another kind of some nice features about it is they do have options for portable models, so it can be taken into the field as well as being used in a traditional laboratory setting, and it is relatively inexpensive comparatively. So these next generation sequencing platforms have allowed scientists to generate just tons and tons of data. What we end up getting are these long reads of nucleotides, and one of the biggest hurdles in the next generation sequencing field is how to interpret this massive amount of data, and that's where we turn to bioinformatics. So how can we take these reads and assemble them into something that can answer questions about what we're trying to study? What do we do with it? So when creating the draft genome, individual reads of DNA are assembled further into contigs, which by nature of their assembly have gaps between them. The next step then is to bridge those gaps between the contigs to create a scaffold. And this can be done using either optical mapping or mate pair sequencing. There's two different types of genome assembly. There's reference-based and de novo. Reference-based genome assembly uses a reference genome to assemble the sequence reads. The reference genome is a complete genome sequence of a known organism, and then the sequence reads are aligned to the reference genome, and the overlaps between the reads are used to assemble said genome. De novo genome assembly does not use a reference genome. The sequence reads are assembled without any prior knowledge of the genome sequence, and this is a more challenging task because it can be used to assemble the genomes of organisms that do not have a reference genome, such as a new exotic species of bacteria or plant or something like that. Reference-based genome assembly is typically faster and easier for obvious reasons. However, it's not always possible to obtain a reference genome for the organism of interest. In these cases, de novo genome assembly is the only option. De novo genome assembly is more challenging to interpret. It makes it difficult to identify genes and other features in the genome. However, it's becoming increasingly more accurate and efficient as the software is becoming more advanced and the algorithms that are used to assemble these genomes are maturing. And it is becoming a more common approach to genome assembly. So some more information on genome assembly. How do we make sure that what we're doing and how we're assembling is the correct sequence? Next generation sequencing platforms by nature do have some inherent error associated with them. So we need to take that into account when we're doing these assemblies. So we're going to start with pre-assembly. This is essential to ensure the integrity of downline analyses such as variant calling or final scaffold sequencing. The steps include two chronological workflows. So first we need to do a quality check. Depending on the type of technology, different errors might arise that would lead to false base call. Sequencing a highly repetitive segment of target DNA or RNA might result in a call that is one short or one more base. And this read quality can be measured and encoded into a score of each nucleotide quality within a read sequence. And then what we need to do with those sequences and the scores associated with them is filter out any reads that failed to pass that quality check. And those then need to be removed from the FASTQ files. And that is in order to get the best assembly of the contigs. Assembly, during this step, the reads will be aligned and utilized with different criteria in order to map each read to the possible location. The prediction position of a read is based on either how much of its sequence aligns with the other reads or a reference. Different alignment algorithms are used for reads from different sequencing technologies. Things like read length, coverage, quality, and the sequencing technique used plays a major role in choosing the best alignment algorithm. And in the case of next generation sequencing specifically. On the other hand, algorithms aligning third generation sequencing reads require advanced approaches to account for the higher error rate associated with them. After we assemble the genome, we have the post-assembly step. So this step focuses on extracting valuable information from the assembled sequence. Comparative genomics and population analysis are examples of post-assembly analysis. A commonly used application associated with Oxford Nanopore Technologies long-read NGS platforms is the Epitome bioinformatics platform. It's a very convenient tool and resource for the analysis of these NGS data. It's designed to be easy to use, even for users with no bioinformatics. And some of the tools that it provides are things like read mapping, variant calling, gene expression analysis, metagenomic analysis, structural variant analysis, and population genetics. It also has a variety of resources, including a database of NGS data, tutorials, things like that, and a nice forum for users to ask questions and get help from other people that are using the software. So as I mentioned a little earlier, here at our laboratory in Madison, Wisconsin, we use the ONT platform for a number of practical applications. We're a high throughput third-party contract testing lab, so we're not an R&D facility, we're not a research university. So we're using this platform in somewhat of a different way than the way it's commonly used in like an academic setting. And that's to answer practical questions for our clients. Actually, starting today, there's the London Calling conference for the Oxford Nanopore technology users. There is a virtual component of that, and I do have a poster that I'm presenting at this conference titled Practical Uses on Nanopore Long-Read Sequencing and a High Throughput Contract Food Microbiology Laboratory. I believe registration is still open and virtual registration is free, so if you're interested in learning more about the Oxford Nanopore platform and interested in this poster, it's being presented virtually over the next couple days. Sorry, I lost my slide here. 1 second. Okay. See if we move on. That'll help. I'll give some more details about how we use that for practical applications later in the talk. Okay, so I want to switch gears a little bit now. We talked about the technology of whole genome sequencing. Now let's talk about some of the pathogens that are can be found in infant formula and the reason that we want to pay particular attention to them. So first, we have Salmonella, very common, well-known strain species of pathogen. It's a rod-shaped gram-negative bacteria. Salmonella enterica is the type species, which is further divided into 6 subspecies. That includes over 2,600 serotypes. Salmonella is named after American veterinary surgeon Daniel Elmer Salmon around the turn of the 20th century. It was first visualized in 1880 in the spleens of typhoid patients. Salmonella is a facultative intracellular pathogen, so it's going to attack cells and cause issues in that way, and it can attack a variety of different cells. It's a very common foodborne pathogen, typically due to unsanitary conditions that can allow it to get into the environment and subsequently into food in a manufacturing environment or in someone's home. Zero tolerance, or the ability to withstand dry conditions, is a trait of Salmonella, and that's due to the presence of a thick, waxy outer layer that helps protect the bacteria from dehydration. It can survive in dry environments for up to several months, which is why it's a particular concern when we're looking at these sorts of dry materials like infant formula, dried milk, other dried products, whey protein, things like that. So how do we test for Salmonella in food? Well, we can look to the FDA Bacterial Analytical Manual, Chapter 5. They give a cultural method for the determination of Salmonella in a variety of foods. There's many different enrichment preparations depending on the food. For infant formula, we're looking at 25-gram subsample into 225 mils of lactose broth. And what this is a general enrichment, so it's going to grow any organism that is present, including the target of Salmonella. That general enrichment is then followed by a selective enrichment, which selects out any non-Salmonella and will continue to grow any Salmonella species or serotypes present. From there, we'll streak those selective enrichments onto a variety of selective growth media. and incubate those plates. If we see growth on those plates, typical growth, then we'll typically take that on to confirmation testing, which also uses a combination of more cultural methods as well as different identification ways to confirm, including whole genome sequencing. There are also many options for real-time PCR rapid screening, and anytime a presumptive is obtained on these sorts of instruments, it needs to be confirmed by a cultural method such as FDA BAM Chapter 5. So here you can see between 1985 and 2005, there were 6 different Salmonella outbreaks in probiotic infant formula across the world. We had a couple in the United Kingdom, United States, some more in Europe and Korea. And this is just to show you that it is a concern, and it is something that needs to be properly managed within a manufacturing facility through environmental monitoring and things like that, as well as end product testing to ensure that the product being released to the market is safe for consumption. Next, I want to talk a little bit about what we've seen more recently in the news. So Cronobacter sakazaki, formerly known as Enterobacter sakazaki. This was first found in a case of septicemia in 1929 and originally called yellow pigmented Enterobacter cloaca. Its first known isolation from a can of dried milk was in 1950, though it's likely existed for millions of years. This is an organism that is ubiquitous in the environment, so it can be found pretty much anywhere, and therefore needs to be, that needs to be noted when dealing with it, not only in a manufacturing environment, but also in people's homes as they're preparing this formula. It was first identified in 1980 by John J. Farmer III and named after Japanese bacteriologist Riichi Sakazaki. It was reclassified into a new genus Cronobacter in 2007 and somewhat disturbingly named after the Greek god Kronos, the titan of Greek myth who devoured his children as they were born. Common with Salmonella is its zero tolerance, so has the ability to survive under dry conditions for very long periods of time. It's a gram-negative opportunistic pathogen. The majority of cases are in adults, but low birth weight, preterm, neonatal, and older infants are the most at risk. In infants, it can cause bacteremia, meningitis, and necrotizing enterocolitis. Invasive infection in infants, while rare, does have a high case fatality rate of 40 to 80%, and that's what makes it a particular concern. As you can see in the diagram to the right, it has a few different areas in the body where it can attack. So the intestinal lumen, thinking about enterocolitis, then it can also pass through that barrier, the epithelial cells in the intestine, and reach the bloodstream. And then from the bloodstream, it can cross the blood-brain barrier and cause meningitis-like issues. So, very dangerous, very important we mitigate its presence, whenever possible. It's commonly associated with contaminated infant formula and other dry materials such as milk, teas, and starches, but has also been associated with things like tampons, and another study found it in 12% of all field vegetables, interestingly. There's also been studies that have shown that it's able to survive in a desiccated state for more than 2 years. So testing for Cronobacter, specifically in food, we're going to look to FDA BAM Chapter 29. There's a real-time PCR screen method for rapid detection that subsequently needs to be confirmed with a cultural method, which can then detect and isolate the species or strain. We can use chromogenic agar for confirmation, as well as things like whole genome sequencing to confirm the identity and the strain present. You can see in the diagram to the right the workflow. So similar to Salmonella, it's a qualitative method. So we're going to enrich a sample centrifuge it down, resuspend it in PBS, and then streak it onto differential growth media or run it by real-time PCR. So just as recently as a week or two ago, Cronobacter sakizaki was in the news. The Washington Post reported on May 11th that a group of epidemiologists are going to recommend to the CDC that they track and have states submit cases of Cronobacter sakizaki, which they already do with Salmonella and more than 100 other pathogens. When I read this article, I was frankly surprised that they weren't already doing this if they are doing it with at least 100 other ones. So they expect this to go through. And again, this is in response to the 2021-2022 outbreak and the subsequent formula shortage, as Keren mentioned earlier in this presentation. Like I said, it could become a federal policy and voted in favor by state epidemiologists and approval from the CDC, and I expect that to happen. So the Cronobacter sakizaki strains identified in sick infants in 2021 did not directly correlate to strains found in the manufacturing facility. And so, something to note in regards to this is that not only does the prevention and the stopping of the organism in a manufacturing setting needs to be of utmost importance, there also needs to be best practices taken by the consumer at home to limit any sort of contamination possibility when preparing infant formula. So you can see on the right, the FDA did put out an infographic on how to prevent Cronobacter illness, giving a bunch of best practices. Because it is ubiquitous in the environment, it can be found, in the lawn, on surfaces. Because it can survive dry environments, it can linger there for a long time. So, proper cleanliness, sanitization, proper storage are all super important to make sure that we don't have contamination of Cronobacter sakizaki in infant formula at the stage where the consumer is using it. One key to prevent this is just to follow the preparation instructions and storage instructions on the label. They've done their due diligence to ensure that all those details are set forth on the label, and they should be followed to every last detail to mitigate any potential of cross-contamination. Okay, so let's tie it all together now. So we talked a little bit about whole genome sequencing and that technology in general. We talked about some of the pathogens that are common in infant formula. So how can we use whole genome sequencing as part of the pathogen confirmation process when we're testing food samples and environmental monitoring samples? So as we mentioned, the FDA put out a letter in 2023 calling for enhanced safety measures to the powdered infant formula industry. So not only did they mention, and they have this paragraph regarding whole genome sequencing and its use, but they also mentioned at the manufacturing facility to do things such as control water in a dry production area, verify the effectiveness of controls through environmental monitoring, implement appropriate corrective actions following the isolation of a pathogen from an environmental or product sample, implement effective supply chain controls for biohazards, and identify biological hazards. All seems like very, common things, but it's super important that all these are in place in a facility so that if it does identify a source of contamination, it can quickly mitigate it. So whole genome sequencing for pathogen confirmations, there's some benefit to WGS and using it to identify pathogens. It has quite a bit of sensitivity compared to traditional methods of pathogen ID, such as culture. It has the ability to identify pathogens that may not be detected by other methods. It can be more accurate than traditional methods of pathogen identification. This means it's less likely to give false positives or false negatives. Speed, so this can be done in real time with next generation sequencing technology, which obviously is more rapid than your traditional cultural identification methods. And this can be very important where that rapid diagnosis is needed, such as in the case of an outbreak. It's very versatile, so it can be used to identify a wide range of pathogens. It's only limited by the databases that can be used if we're going to be doing a reference-based assembly. So you see I have the NCBI logo there, which is a common place to go when looking for reference-based assembly. And then finally, cost. As I described earlier, whole genome sequencing cost has dropped dramatically over the past 2 decades, making it much more affordable to be used in clinical laboratories or research institutions. The infographic schematic on the right is from the CDC and kind of gives a breakdown of the process of whole genome sequencing. So we have a culture of bacteria. What we'll do then is we'll take and purify the DNA. We'll shear the DNA into fragments. We can use the fragments to amplify a particular target, and then that library is loaded onto the sequencer. And those combination of nucleotides can then be analyzed using specialized computer programs like I described earlier in this webinar. So another key benefit with whole genome sequencing for pathogen identification, and this kind of harkens back to the FDA call to the infant formula industry for the use of WGS, is its ability to track strains through a production environment. And the way this is done is we can collect samples from a suspected source of contamination or just through routine environmental monitoring. We'll extract the DNA, sequence the DNA, and then compare that sequenced genome to a database such as NCBI. And that way we can identify the pathogen strain. And then from there, we can track that strain throughout the facility. It does this by comparing the genomes of isolates from different sources. If the genomes are similar, then it's likely that the isolates came from the same source. If the genomes are different, then it's likely that the isolates came from different sources. So things and benefits for the use of WGS for strain tracking help identify the source of contamination. Pathogen tracking can help identify that, which can of course help to prevent future outbreaks. We can track the spread of that contamination, which can help identify and isolate contaminated products, and also monitor the effectiveness of interventions. So, typically that involves thorough cleanup and disinfection, sanitization procedures, employee training, things like that. And using WGS to look at strain level identity can help monitor that effectiveness of the corrective actions being put into place. And what does this lead to? Well, it leads to improved food safety, reduce food recalls, increase consumer confidence in your brand, and reduce costs, of course. I mean, these sorts of events can cost companies millions of dollars in product and losses and litigation and things like that. So it can be a super important and beneficial practice to put into place. And of course, it's recommended by the FDA at this time. So just a little bit about the service offering we have here at the Madison Laboratory. We do have, like I said, we do have a whole genome sequencing service offering as part of a routine test option for clients looking to get a fast and affordable way to obtain a genomic identification for their product of interest. Pathogen confirmation identification testing is part of this offering. We can sequence isolates from a presumptive plate, from an enrichment, or directly from the sample itself. Like I mentioned earlier, we use Oxford Nanopore Technologies GridION platform with reference-based genome assembly, and which can provide reliable species-specific confirmation identifications. And we do have the option for strain-specific and strain tracking available upon request. So, as I mentioned, and I get into a lot more details in the poster I'm presenting at the ONT London Calling Conference. We use this technology for a variety of different practical applications, not just pathogen confirmation. So we can look at things like metagenomic identifications of blends of bacteria, such as probiotics or starter cultures, microbiome characterization of fermented foods, such as kefir or kombucha. You see a picture of a kombucha product on the upper right there. We can look at spoilage investigation. So, does your product appear spoiled? Does it have bloating, like the picture in the middle there? We can use whole genome sequencing to investigate the organisms present in a much more comprehensive way than we could with traditional cultural spoilage investigations. And then, looking at non-confirming presumptive results from rapid screen tests and helping troubleshoot why we're seeing those discrepancies between what the instrument is giving and what's showing up or not showing up on the cultural plate when we're confirming that presumptive head. In summary, it's a really powerful investigatory tool that can be used to answer a multitude of questions related to bacterial presence and identifications in a wide variety of matrices. And with that, I'd like to thank you for attending and listening to our webinar today. You can see I put a couple contacts here, so Keren's email, my email, and my colleague Dave's email. Any questions regarding infant formula can be directed to Keren. Whole genome sequencing questions, Dave and I. So look forward to hearing from you. And we do have some time, I believe, for some Q&A right now. So again, thank you very much for your attention.
Great, thank you for the presentation. Now we will move into a short Q&A. Feel free to continue to submit questions during this session. If we do not address the question that you asked live during the presentation, we will follow up with you directly after the webinar. So the first question is, what is the turnaround time for results by sequencing at your lab? And then the same person asked, how are these results kept confidential?
That is a good question. So as I mentioned, our whole genome sequencing offering is a routine test. We have test codes for this, and turnaround time is typically around a week, though we do have options for rush as needed. And then, like everything we do, it's kept strictly confidential. So the sequences that we're obtaining are stored in a secure database. It's not public information by any means. So just like any other results we produce, we hold confidentiality to the highest extent.
Awesome. Next we have, how do you ensure that your comparison database is up to date and contains the most recent and relevant species or strains?
Sure, yeah. So that also is a good question. To ensure that we have the most up-to-date information, we do rely on the curation of the databases used, which are commonly updated, or I would say with regular intervals and regular frequency, as new strains are identified, as things are cleaned up and deemed more accurate. And then from there, if there's any discrepancies between what we're seeing in our curated database with what the instrument is producing, then we can go back to NCBI and blast against the NCBI database to confirm, yes, this is what we're seeing. And the NCBI does have some level of curation in the ref-seq portion of the database, which is where we commonly go to.
Okay, next we have, out of all of the different instrument options you described in your talk, why did you choose the Oxford Nanopore system?
I mean, we get that question on a regular basis. As I mentioned, there's a lot of different options available. They all have different benefits and drawbacks associated with them. Oxford Nanopore, it really gives us a lot of nice flexibility. So we have the option to run as little as one sample or as many samples as we want at a time in order to meet those rapid turnaround times A common thing in third-party sequence testing is to have very extended turnaround times for any reasonable cost. You know, you're looking at weeks to result because the consumables are very expensive, and the laboratory wants to fill a flow cell, for example, in order to make sure that they're meeting their margins when it comes to testing. So, you know, for those clients that don't and can't afford to wait 4 weeks for a result, Oxford Nanopore GridION allows us to rapidly and efficiently provide results, like I said, within that 1 week turnaround time, and gives us that flexibility that we wouldn't necessarily have with another platform. And I think that really sets us apart from a lot of other laboratories that have this service offering, is that speed to result.
And we're going to do one more question, and this is definitely more technical. Is whole genome sequencing considered as next genome sequencing, or are they two different technologies?
Yeah, so they're different in the respect that I think whole genome sequencing is like a science. So it's the practice of taking and assembling a genome of a particular living thing or organism. Next generation sequencing is a way to perform whole genome sequencing. So, you know, as I mentioned early in my talk, you know, kind of the advent of sequencing, of whole genome sequencing, was Sanger sequencing, very slow, but very accurate and effective, also very costly. Next generation sequencing has taken an approach that is much faster and more efficient, and there's different technologies to perform that, but it's more of the case of next generation sequencers, and next generation sequencing is a type of whole genome sequencing, would be a good way to describe that.
Okay, that wraps up our Q&A session. I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available in 3 business days. Thank you for your expertise and presentation today, and thank you everyone for attending.
Thank you all.